Okay, welcome. I'll just add to Jeet's welcome. Happy to have you all here. And my role today is to talk a bit about why you should care about glycans. And I thought the best way to do that would be really just to start from the beginning. That seems like the best place to start, and start with this complex this concept that's essential for glycobiology, and that is that all living organisms produce complex carbohydrates of one type or another. So here's a very simple single cellular organism, a fungus. In this case, it's yeast, and you can see here is the actual organism, and all of the coat around this clear area is all complex carbohydrate. And this organism places this coat around it to protect itself from its environment and to regulate what molecules it sees within its environment. So this coating of carbohydrate is made up primarily of glycans or glycoconjugates, and these turn out to be absolutely essential for all kinds of biological functions that occur at the cell surface. And it's especially true for protection, for cell signaling, and for cells to be able to recognize each other. We also appreciate now in the last decade or so that there is glycosylation that happens within the cytoplasm and within the nucleus, so-called nucleocytoplasmic glycosylation, which we'll talk about in a bit. But here in an animal cell, and this happens to be a human blood cell, which has been stained with the dye that shows all the carbohydrates at the cell surface, you can see that there is a rich coating of carbohydrate at the surface of all animal cells as well. And this is sometimes referred to as the glycocalyx, almost as if it is its own organelle. And in many ways, it is. It functions in the same ways that the fungus glycocalyx functions in protection, in signaling, and in cell-cell recognition. So for the sort of introductory session, there's some learning objectives that I've stated here, and that's to understand the scope and breadth and the impact of this field that we call glycobiology. And some of this I think many of you already know, but I think it's best just to start from point zero, and we'll build from there. We'll talk about how structural diversity of glycans linked to proteins and lipids arises from the chemistry of their building blocks, and the diversity of the way that the building blocks can be put together. We'll talk about the different classes of glycoconjugates that are found in vertebrates. And as we talk about those, we'll speak a bit about some of the functional impacts if you lose those classes of glycoconjugates, and also some strategies for understanding how the glycan diversity is understood by the organism and utilized by the organism. At the very end, I will just sort of segue into Raja's talk to follow and try and place glycoinformatics in the context of other informatic efforts in biomedical research, and a little bit of sort of high-level thought about how we might connect, or what's valuable about connecting our knowledge to knowledge in other fields. So let's start with why you should care about glycobiology. So here I've listed some of the more well-known diseases and conditions that have been linked to the expression of particular classes of animal glycans, and some of these I'm sure you're aware of, but starting at, really sort of a very essential component of how our bodies work, the major ABO blood groups in humans, these are all antigens that are glycans found on the surface of cells, especially red blood cells. In fact, all of transplantation biology and transfusion biology would be a big black box if we didn't really understand how these glycans mediate the ability of our bodies to recognize self and non-self. So essential for all of human physiology. We know that glycans and binding proteins that recognize those glycans are found on the surface of many different types of inflammatory cells, neutrophils, eosinophils, macrophages, lymphocytes, and these glycans and the binding proteins that recognize them are important for determining the severity of many sorts of chronic diseases, such as asthma, irritable bowel disease, COPD, and also autoimmune disorders like lupus, rheumatoid arthritis. So also we know that cancer cells have a very different sort of glycan surface that they put out for the rest of the world to see. And as cancers become more and more metastatic, the cells and tumors frequently change their glycans, and these changes frequently allow cancer cells to move away from the tumor, move to other tissues in the process called metastasis. There's a bunch of other diseases that also have associated with them altered glycan expression, and I've listed some here in which altered glycan expression causes or has been linked to or provides therapeutic targets for diseases such as diabetes, congenital muscular dystrophies, lysosomal storage diseases, mucopolysaccharidoses. Neurodegeneration is a broad sort of pathologic process. X-linked intellectual disability, sickle cell disease, coagulopathies, stroke, and host pathogen interactions, both viral and bacterial, and therefore sepsis. So it's been said by people that think a lot about what happens in this field that almost every human disease is in some way tied to glycans or changes in glycan expression, especially because they're so important in inflammatory disorders. So these are sort of pathogenic conditions, but glycans, we have them for a reason, and the reason actually is because they are exquisitely altered in their expression in relation to tissue architecture, tissue structure, tissue function. So if I remind you again about the glycocalyx, we know, and I'm going to show you, I think, a particularly provocative example of how the glycocalyx differs from cell to cell, even in one tissue. And if any of you have heard me talk before, I frequently use this slide as an introductory slide because it just always still excites me about how little we know in glycobiology. And so this is a section of a rat brain that's been stained with 3 different antibodies. Each of those antibodies recognizes the same glycoprotein. They just recognize different glycoforms of that glycoprotein. That is the same polypeptide backbone, but with a different carbohydrate structure on particular sites on that particular glycoprotein. So on the upper left here on this panel, you can obviously appreciate that within the brain, this glycoprotein is being glycosylated differently in different parts of the brain in sort of a global sense. So you can see here in the brainstem, the blue form of this glycoprotein predominates. In the hippocampus and cerebellum, you begin to see some reddish forms here in the cerebellum of the glycoprotein. And if you look up here in the cortex, you're seeing a different sort of diversity of this glycoprotein expression. And in fact, if you zoom into the cortex, which is shown over here on the right, and you can see now the laminar structure of the neurons in the cortex, you can see that even within one layer of the cortex, cells right next to each other might be glycosylating the same protein in a different fashion. So this glycoform heterogeneity exists even at the level of single cells within a tissue. And this sort of poses some, I think, very important questions to think about. And those are, so what are the functions that are subserved by having this cell-specific glycan presentation, even at the level of single cells and differing glycosylation on neighboring cells? And how does this sort of obviously very finely regulated glycan expression fit within the conceptual framework that we've all been taught that says regulated gene expression controls biology? How do these glycans fit within our concept of how cells and tissues work? And so I'll back up a little bit here to the central dogma, which we all learned about as we were being trained, which said, of course, that DNA encodes RNAs that can be translated into proteins, the central dogma. And in fact, those proteins work together to feed back on transcription and translation and transcription factors, so they regulate their own production and come together in a cell to form a functioning cell, and cells come together to build organisms, right? So that's sort of the central dogma, but this dogma really very much ignores a role for lipids and carbohydrates, and especially for things that are happening at the cell surface in order to build organisms. So I propose that the more expanded and more useful dogma of life is that, yes, DNA encodes RNA, which is translated into proteins. Proteins work together to form functioning cells that can build organisms, but also some proteins are enzymes. Enzymes build glycans, build lipids, build glycoconjugates. The glycoconjugates form the interface between cells that allow cells to build organisms. And in fact, we now know that many of the proteins that are transcription factors that regulate transcription and translation are also modified with carbohydrates, and that those glycan modifications on transcription factors have very important consequences for their function, just like phosphorylation. So glycans are all over the central dogma of life. And to turn to sort of a more practical concept of why glycans are important, I'll just point out that essentially all biologic therapeutics are glycoproteins or glycoconjugates, right? So this slide's a bit old now. This is has this increase in the market size for biologics. By that we mean therapeutic proteins, monoclonal antibodies, vaccines. This market has continued to grow certainly well beyond 2017, but the point here is just to appreciate that almost all actually of these biologics are carbohydrates, and those carbohydrates have important roles in the efficacy of those drugs. And I'm going to give you one particular example that's been really well thought out and characterized. So erythropoietin is really a textbook example of how important glycans are for drug efficacy. So here's a model of the erythropoietin protein with the protein backbone shown in blue in the ribbon diagram. And then modeled on top of that protein backbone are glycans, here shown in red. And these are N-linked glycans, linked through asparaginins on the protein backbone. And erythropoietins are relatively small proteins. So in this case, the glycans are almost as big in terms of the space that they subsume as the protein backbone is itself. And so if you purify erythropoietin from human tissues, it has 3 N-linked glycans on it. Sometimes it also has an O-linked glycan. So this 18 kilodalton molecular weight protein backbone is actually 34 kilodaltons with all the glycans put on it. If you express erythropoietin in CHO cells, which one company called Amgen did quite a while ago, it's purified from these CHO cells with 3 N-glycans and 1 O-linked glycan. And this is Amgen's drug Epogen, which was just a world changer for that company, and was introduced in response to a need for anemia therapies, especially in patients with kidney disease, where they chew up their red blood cells and they need to have enhanced red blood cell production, which is what erythropoietin does. So Amgen developed recombinant erythropoietin called Epogen and introduced it in the market, and it just really took off. And it became a very, very powerful marketed drug and really helped out in kidney disease and also in important applications in chemotherapy for cancer, where red blood cells can be, where anemia is a hallmark and red blood cells can be reduced. Right, so it was a great drug, but it turns out that the engineers at Amgen started playing around with their drug, and they knew that looking at the efficacy of the drug here, this is native Epogen with 3 N-linked glycans, if they added additional N-linked glycan sites to the Epogen molecule, the drug became more and more potent. And I want you to appreciate that these are log increases in the potency of this drug. And so for a drug company, this is great. It means less drug is needed, so cheaper cost of products and more potent drug. For patients, it means less drug that needs to be taken, hopefully less chance for side effects. And Amgen settled on a form of erythropoietin they called Aranesp, which has 5 N-linked sites. And it was commercialized and absolutely, upon its commercialization, just rocked this market. Amgen essentially doubled their revenues from the drug. And in fact, this is a story for another time, but the drug was so potent that people began to see side effects that caused drug doses to be backed off on, but still is an important therapy for various forms of anemia. Right. So in terms of basic biology, in terms of applied biology and therapeutic markets for biologics, glycobiology sits at the core of making better drugs and making better tissues. So let's talk a little bit about the basics of what glycoconjugates are, and I'll introduce some terms that I'm sure many of you have heard before. The first is the building blocks of glycans, or complex carbohydrates, are monosaccharides. And a monosaccharide is essentially a carbohydrate that cannot be broken down into any sort of smaller unit. And usually the gold standard for this is acid hydrolysis. What you get following acid hydrolysis of a glycan is the monosaccharides. We put together 4 to 12 of these monosaccharide units you get an oligosaccharide, and different people have different cutoffs where they think you go from an oligosaccharide to a polysaccharide. But in general, once you get to something with more than 12 monosaccharides, you're beginning to think about something in the polysaccharide world. And these polysaccharides that can be very large are often long linear repeating chains that may have a single monosaccharide or a repeating disaccharide motif that build up these long polymers, and sometimes there can be branches off of those polymers with other monosaccharides or other chains. Okay, so for glycoconjugates, we have glycoproteins, which are obviously proteins plus carbohydrates. And we've mentioned that most of the proteins of biological and therapeutic significance as drugs are glycoproteins. Glycolipids are lipid plus carbohydrate. These are important tissue and cell type markers. Most of the ABO blood group antigens exist on proteins, but also as lipids, especially on red cells. And these are also interesting tumor-associated antigens in many sorts of cancers. And then there are proteoglycans, which are a complex of a protein, again, like glycoproteins, but then they have a different sort of carbohydrate attached to them called a glycosaminoglycan. And these are polysaccharides that can be quite large with a characteristic repeating disaccharide motif. And these have immense structural diversity because these glycan backbones can be modified post-synthetically with sulfation and other sort of chemical modifications. And they have a lot of different functions, especially in terms of tissue stability, extracellular matrix functions, growth factor presentation, cell signaling. So a very pleiotropic family of glycoconjugates. So when you think about proteins, we think about the 20 amino acids plus 1 or 2 that build all the proteins in our body. For building glycans, we have a smaller set of building blocks. And the ones I'm sure you recognize: glucose, the N-acetyl form of glucose, N-acetylglucosamine; galactose, the N-acetyl form of galactose, which is N-acetylgalactosamine; mannose. And then we have ones you may not have heard about as much: xylose, glucuronic acid, fucose, and then finally a very important family of monosaccharides that tend to cap other oligosaccharides, sialic acids. And this is one of those sialic acids. This is N-acetylneurominic acid. But the sialic acids are a family of structures that have different sorts of modifications in various parts of the monosaccharide backbone. Right, so these monosaccharides, we have a relatively small number of building blocks, but unlike polypeptides, where the amino acids are linked together in only one way to form a linear polymer, monosaccharides can be put together in various different ways. And here we're looking at a molecule of lactose, which is a disaccharide of galactose linked to glucose. And to form lactose, that linkage between one monosaccharide and the other is from the 1 position of the galactose to the 4 position of the glucose. And the linkage has a stereochemistry, which we call beta. It can also exist in an alpha form stereochemistry, where this link would be not up to the top, but down to the bottom. So even in terms of thinking about how 2 monosaccharides can be put together, you have alpha versus beta anamericity, but then you can have linkage between these two. Here's to the 4 position, but also to the 3 position, or the 2 position, or the 6 position. Wherever you see a hydroxyl, you can link theoretically between those 2 units. And then in these other linkages, it won't be lactose, it'll be some other disaccharide. But you can see how even with 2 monosaccharides, you can begin to build a number of different molecules that have the same chemical composition, but different stereochemistry. And so just kind of to put an exclamation point on this, if you think about proteins, you have 20 amino acids, and you can put those, if you took 3 of those amino acids, you could put them together in 6 different ways, right? And nucleic acids are the same thing. Here you only have 4 building blocks, and you have, if you try and make a trimer, you can make it in 3 different ways. Now with monosaccharides, if you pick 3 monosaccharides, because of the linkage position heterogeneity and anamericity, you really get an exponential growth in the structural possibilities of just putting together 3 different monosaccharides. And then if you layer on top of that different sorts of post-synthetic modifications like sulfation, the potential to build structural diversity within this family of molecules just becomes immense. Now, not all of these structures exist. Structural heterogeneity is restricted by the enzymes that we have to put these things together. But as you look across organisms, you begin to see more and more sorts of glycan structures that begin to fill out this sort of level of complexity. And as I've mentioned, once you assemble these things together, you can actually modify them post-synthetically with different sorts of secondary modifications. Right, so thinking a little bit about how these things are made, unlike when we think about DNA synthesis, RNA synthesis, protein synthesis, which are driven by templates, glycan structures are not encoded in that way. There's not a template for building a carbohydrate. So the correct synthesis of these glycans depends on the expression of the right enzymes that build the polymers, the expression of enzymes that can trim those polymers back so that they can be modified in a different way, the availability of the building blocks, these monosaccharides, that have to be activated into sugar nucleotides before they're accessible for the enzymes to use, and also where these enzymes are placed within the cell. They don't float around in the cytoplasm. They actually exist within organellar subcompartments. And the enzymes and the substrates all have to be brought to the right place at the right time in order to build an oligosaccharide on a protein or a lipid. Many of these steps require high-energy phosphates, either as nucleotide donors or for transporting substrates and molecules across the cell. So the energy charge of the cell, the nutritional state of the cell, all of these things can contribute to what kinds of glycans are found on the surface of a cell. So glycosylation is very sensitive to subtle shifts in the environment. All right, so some basic terms related to glycoconjugates. The important concept is that of microheterogeneity. So this is the variation seen in glycosylation at a given glycosylation site on a glycoprotein, for instance. The site on a glycoprotein may be unoccupied, or it may be occupied. And if it's occupied, it may be occupied by different sorts of glycan structures. Even coming from the same cell at the same time, the population of a particular glycoprotein that's made may have heterogeneity at the sites of glycosylation. So this led a lot of biochemists in the 20th century, mid-20th century, to think that, well, maybe glycans aren't that important because they're so higgledy-piggledy and heterogeneous from protein to protein. I've already touched on the concept of glycoforms when I showed you that rat brain, and that is that a glycoprotein or a glycolipid can exist with different glycoforms. That is, a non-carbohydrate portion may remain the same, but variation in the carbohydrate portion can create different glycoforms of a glycoconjugate. And finally, you'll hear this term thrown around, glycone versus aglycone. In general, the glycone of a glycoconjugate is the carbohydrate part, and the aglycone is a non-carbohydrate part. And really, this is a chemical term, thinking about maybe a carbohydrate linked to an amino acid. So the amino acid would be the aglycone, and the carbohydrate would be the glycone. But it's been expanded to think about not just an amino acid, but also the whole protein or the whole lipid as the aglycone. Okay, so that's sort of a primer on sort of the basics of thinking about glycan structural heterogeneity and how it arises. So I'm going to turn now to walking through some of the classes of we'll actually touch on all the classes of glycoconjugates found in vertebrates, and a little bit about their functional impacts, and a little bit about how to understand what they do. And this is a diagram that you go to seminars in glycobiology, you'll see forms of this diagram over and over and over again. And so it's a good place to introduce the different types of carbohydrate structures that you see on animal cells. And here's represented the plasma membrane of a generic animal cell, and a glycoprotein with a transmembrane domain here, and different forms of glycosylation placed on the polypeptide backbone of the folded glycoprotein. Right, so we'll touch base a little bit on different classes of glycans or glycoconjugates. So there's GPI anchors, which serve to anchor some proteins to the cell surface, This anchor is linked to a phospholipid embedded in the plasma membrane, and that forms sort of the, if you will, the transmembrane domain, although it's not transmembrane, the membrane anchor for subsets of different glycoproteins. There's GPI anchors, where the anchor is made of carbohydrates. And then there are the glycans that you found more generically on glycoproteins. So these include the classic types of N-linked glycans, where the glycan is linked to an asparagine. These come in different flavors, so-called high mannose, hybrid, and complex. We'll come back to distinguish those in a minute. Then there are the O-linked glycans, which are found where the glycan is linked to a serine or threonine residue. And these have different sorts of structural classes that we'll touch on a bit. Then there are some glycans, usually O-linked, that are found on particular domain structures on proteins, say on IG domains or thrombospondin repeats. They tend to carry specific glycan structures that have their own classes. And some of these are O-glucose, O-fucose, O-mannose. Okay, so there's the N-linked and O-linked glycans on glycoproteins. There's another class I've mentioned of O-linked glycans called glycosaminoglycans that are linked to serines and threonines. These are the long, generally long polymeric repeats disaccharide structures that can be heavily sulfated in certain contexts. And then our one example of a soluble polysaccharide in animal cells, and that's hyaluronic acid, which is found throughout the body. It's very abundant glycan, but especially concentrated in joints and connective tissues. Right, and then we have, as I've mentioned before, glycosylation that happens in the cytoplasm and the nucleus. And there's sort of two examples of this that are important to remember. One is the addition of N-acetylglucosamine onto serines or threonines of many, many cytoplasmic and nuclear proteins. Akin to phosphorylation, it's dynamic, it goes on, it comes off, and has functional consequences for how that polypeptide carries out its activities. Also, we should never forget about glycogen, a storage energy form of glucose, which is attached to a protein in the cytoplasm called glycogenin. Right? And I wanted to make sure to point out, also point out that collagen is a glycoprotein. Collagen has its own special type of small glycan, a disaccharide usually, on its polypeptide backbone on lysines that have been hydroxylated enzymatically. And these glycans are essential for collagen to form its helical structure. And then glycolipids, which we'll talk about as well, that have structural heterogeneity in the carbohydrates that are linked, in this case, to ceramides, not to glycerol backbone lipids in the plasma membrane. But you've seen across this diagram these little shapes, different colors, circles, squares, diamonds. These have meaning. And over the last 10 to 15 years, the field has settled on representations that when we see, we recognize immediately what that representation is. So mannoses are always green circles, galactoses are always yellow circles, unless they're N-acetylated, and then they're a square. Same for glucose, always a blue circle unless it's N-acetylated, and then it's a blue square. If it's amidated but not acetylated, it's a half blue square. So this would be glucosamine. This is N-acetylglucosamine. And then there are the sialic acids and other acidic sugars like glucuronic acid and iduronic acid. Fucose is always a red triangle. So these are things that you immediately recognize and are a shorthand way for us to represent structures without having to draw them out. But it's important to remember that the function of these molecules really is based on their true three-dimensional structure, not on in space, not on the color that they happen to have in our shorthand. So I'm going to start now with N-linked glycans and talk a little bit about their biosynthesis and their structural heterogeneity. Right, so we're looking here at the endoplasmic reticulum in this rather at first maybe daunting image. So this is the lumen of the endoplasmic reticulum. This would be the cytoplasm out here. And we're just marching through looking at how messenger RNA that encodes for a protein that's going to enter the secretory apparatus, sits on the rough endoplasmic reticulum. The protein is translated and inserted into the endoplasmic reticulum. If that protein has a consensus site on it for end-link glycosylation, and that consensus site is pretty well understood to be an asparagine, and then any amino acid other than proline, and then a serine or a threonine. And we now appreciate also cysteine can be in that third position. So asparagine X, ST, or C, if that consensus exists, it will be recognized by an enzyme called the oligosaccharyl transferase that will transfer in block a very large glycan consisting of 2 N-acetylglucosamines, 9 mannoses, and 3 glucoses. It just gets chunked right onto the polypeptide backbone as it's being translated. So how do you get to the point of having that large glycan? Well, it builds up beginning on the cytoplasmic face of the endoplasmic reticulum, requires a complex long lipid called a dolichol that has to get phosphorylated, and then it gets glycosylated to form, in this case, glucnac pyrophosphodolichol, and that dolichol with one sugar gets built up sequentially by different enzymes that all act on the cytoplasmic face, and each enzyme requires a nucleotide sugar to donate the monosaccharide, and these get built up to a certain size, a well-characterized size. In fact, the MAN5, so 5 green circles and 2 blue squares, MAN5 glucnac2, which gets flipped into the endoplasmic reticulum, still on the dolichol, and then gets extended out by new enzymes to this MAN9 gluc3 glucnac2 structure on a dolichol that the oligosaccharyl transferase takes and places on the polypeptide. Subsequently, this large structure, beginning in the ER and then in the Golgi, will be chewed back by enzymes that sequentially remove one or more of these monosaccharides down to a different structure that can then be built out again to generate the structures we call high mannose, hybrid, or complex. So you start with this large glycan in the endoplasmic reticulum that gets chewed back and then re-extended to generate the various structural diversities. But I want you to appreciate, before we move out of the ER and look at other sorts of N-glycans, all the different sorts of metabolic pathways that feed into this process, right? We have lipid metabolism, isoprene metabolism to build the dolichol. We have nucleotide metabolism to provide the nucleotide sugars. We have sugar metabolism to provide glucose and convert that glucose into other monosaccharides by various biosynthetic pathways. So sugar metabolism, lipid metabolism, nucleotide metabolism, and amino acid metabolism as well all feed into the production of the glycoproteins. Right, okay, so I've thrown these terms around: high mannose or oligomannose, complex, hybrid. These are different types of N-glycans. So in the endoplasmic reticulum, we saw that a high mannose glycan with 3 glucoses is added to the glycoprotein. Those glucoses are rapidly removed as the protein folds. Once the protein is fully mature, it may still have a high mannose glycan on it. Transitioning from the ER into the Golgi apparatus, many of these mannoses can be removed, frequently down to this MAN5 structure, which still retains some high mannose character on one arm of the N-glycan, but on the other arm, it may be chewed down all the way and then extended by N-acetylglucosamines, galactoses, and may be capped by sialic acid. So this structure that has some characteristic of the high man glycan and some characteristics that are more complex than that, these are called hybrid glycans. And these may stay that way and end up out at the cell surface as hybrid structures. If all of the mannoses get chewed down, so you only have the trimannocele core, and then the trimannocele core gets extended, this becomes a complex glycan, which has maybe 2 arms. There could also be other branches off of these mannoses to give up to 4 or 5 arms. You can also add an N-acetylglucosamine to this internal mannose to form what's called a bisected structure, which can add another N-acetylglucosamine, which usually does not get extended, but just remains as an N-acetylglucosamine. There's another process called core fucosylation, which adds a fucose here, the red triangle, to the internal N-acetylglucosamine, to form a fucosylated glycan, and you can have fucosylated hybrid, fucosylated complex, and yes, even fucosylated high mannose glycans on asparaginins on glycoproteins. And these are important. And I'll just show you one of the first examples that made people appreciate that the particular type of glycan you have are able to make is essential for life. And that is the fact that the loss of hybrid complex glycans, that is, if an animal is only able to make high mannose glycans, that results in embryonic lethality. And this is a paper published by Pam Stanley a couple decades ago that showed that if you lose this enzyme, N-acetylglucosaminotransferase transferase 1, GLUCNAC transferase that adds that blue square onto a glycan core to make hybrid and complex glycans. If you don't have that enzyme, you're not healthy. And this mouse, I think around day 14, this mouse arrests and stops developing. You can see it's smaller and it's actually not real healthy and will die. So complex glycans are essential for life in vertebrates. Okay, so that's end-linked glycans. O-linked glycans have their own structural diversity that's different from the end-linked glycans. O-linked glycans are especially prominent on families of proteins that have mucin-type repeats or mucin-type domains. So here's sort of a cartoon of a mucin-type protein where it has heavy abundance of O-linked glycans, and this correlates with the density of serines and threonines within this backbone. In fact, there's islands of serines and threonines frequently separated by proline residues. So proline ser3 kind of motifs that can be identified in these mucin-type domains. And each of those serines and threonines can be modified with different types of O-glycans. All of these mucin-type O-glycans begin with an N-acetylgalactosamine, the yellow square, onto a serine or threonine. And then depending on how they're branched and extended, they can be different sorts of cores. So this is core 1. Core 2 is core 1, but it has an N-acetylglucosamine linked to the 6th position of the GalNAc. This is core 3, which instead of a galactose here has a GLUCNAc. And then core 4, which instead of having a galactose has 2 N-acetylglucosamines. And these structures can be identified chemically by various sorts of approaches. I mentioned before that there are other types of O-glycans, O-mannose, O-fucose, and O-glucose. And these are generally found on specific types of domains, protein domains, just like the mucin type that tend to be found on mucin repeats, although they can also be found in other serines and threonines that don't exist within a mucin domain. So why are O-glycans important? Well, we appreciate that O-glycosylation on mucin-type proteins changes rather dramatically in several types of cancers, especially the addition of sialic acid, in particular linkages in particular sites in the glycan can have important consequences for how the tumor cell is recognized by the immune system. This company, Paleon Pharmaceutical, raised a huge amount of money not that long ago to figure out ways to remodel the glycans on mucins in order to hope that those cells might be more amenable to killing by the immune system. So glycolipids, another major type of glycoconjugate. The glycolipids are glycans linked to a lipid called ceramide. Well, ceramide is a bit different than the phospholipid backbone. And most of the glycolipids begin from ceramide by the addition of glucose to form glucosylceramide. And that glucose then is capped with the galactose that can then be extended in various ways. And what I'm showing here is the family of glycolipids called gangliosides. Depending on which monosaccharides you add after this glycose, you may form a different family of glycolipids. The gangliosides are very important in nervous tissue. They're highly enriched in nervous tissue. These glycolipids also serve as receptors for bacteria and toxins, so that's not good, but we've retained these glycolipids because they're really important for other aspects of our normal development and tissue function. This enzyme called ST3-gal5, which adds sialic acid to lactosylceramide, forms this very simple ganglioside called GM3. So this is GM3 synthase, also called ST3-gal5. So this enzyme was knocked out in mice about 2.5 decades ago. And surprisingly to everybody, the knockout of that enzyme in mice gave you a viable, fertile mouse that actually, in some ways, was a bit healthier. It was resistant to forming of getting type 2 diabetes. These mice had enhanced insulin receptor signaling. They did have loss of hearing due to death of cells in the cochlea, and they had slightly reduced sperm production. But overall, the mouse was lethal, was viable and fertile, which made people working on glycolipids really disappointed because they thought these glycolipids were going to be really important. And at least in mice, they weren't. Well, about 10 years ago, mutation in ST3-GAL5 was identified in humans and was sort of at the same time identified both in a population of African Americans in South Carolina. And I'll mention them first. And this was this syndrome that became known as GM3 deficiency, was originally called salt and pepper because the clinical presentation was in sort of scattered hyper and hypopigmentation in the skin. The family that this was first identified in, the kids had really a profound intellectual disability. They were born completely normally, but within a month or two, their head stopped growing. They developed a very severe seizure disorder, and most the kids didn't survive past 8 or 9 years old. At the same time, this mutation in the same enzyme was identified in the Amish community as a relatively abundant mutation with very, very, very similar clinical presentation, especially in regards to seizures and ability to thrive. The mutation is in the same enzyme. It happens to be at a different site. And we actually now know that there are about 6 or 7 other cohorts of mutations in this enzyme in other groups around the world, and all have essentially the same clinical presentation. So in humans, loss of that enzyme and loss of the ability to make all those glycolipids is actually really essential to life. Okay, the last form of glycoconjugate I'm going to introduce are the GPI anchors. Remember, I mentioned that GPI anchors link some glycoproteins to the plasma membrane through a phospholipid backbone, right? And that phospholipid is a phosphoinositol. So here you see the inositol ring, which gets an N-acetyl, which gets a glucosamine added to it, and then 3 mannoses, and then an ethanolamine that becomes the link, the bridge between the glycophosphatidylainositol and the protein. This glycophosphatidylainositol is built up in the endoplasmic reticulum, and then through a transaminase reaction that involves several different polypeptides, cleaves the polypeptide and adds it to the ethanolamine of the GPI anchor. There's not a strict consensus to where this happens, but there's sort of certain features on the polypeptide backbone that are conserved, and that is the presence of some small polar residues close by the cleavage site, followed by a stretch of hydrophobic residues. A lot of different proteins have the sequence in them, and many proteins that exist as transmembrane proteins can also exist as GPI anchored proteins. This allows the cell a way to quickly release these proteins. So many of these proteins are actually adhesion molecules, and there is a GPI specific phospholipase that can cleave this anchor in the phospholipid part of it and release the protein. Right, so these are important. So here's one example of when GPI biosynthesis goes awry. There's a very important protein on red cells called CD59 that's GPI anchored. If this GPI anchor biosynthesis is disrupted, the protein is not presented at the cell surface, and normally this protein protects red cells from being lysed by complement. So in the absence of this GPI anchor biosynthesis, red cells lyse to give a disorder called paroxysmal nocturnal hemoglobinuria, where red cells lyse. And you can see these are urine collections taken from a patient starting in the morning. And there's some variability across the day, which isn't really clear why, but not blood, but the breakdown components of red blood cells accumulate in the urine, and eventually in the kidney tubules, which are shown here, which results in loss of kidney function and is a major problem in this disorder. Okay, so in the last couple minutes, I want to turn to, first of all, a strategy for trying to understand how cells harness all of this structural diversity, and then a little bit about informatics associated with glycans. So we've talked about glycans on animal cells. Here's shown sort of a little itty bitty, maybe O-link glycan with or a glycolipid even with the galactose and acylic acid. And many of these glycan structures found on cells are recognized by glycan binding proteins, shown here in blue, which have structural specificity for particular glycans. So this is one way that an immune cell, for instance, can recognize another cell as self because it has the right glycans on it to be recognized by the glycan binding proteins. And that binding activity might have consequences. Turns out also that pathogens have understood how important these glycans are to us, and many pathogens have their own glycan proteins that recognize glycans on our cells and use those glycans as receptors to gain entry into our tissues. Also, not just pathogens, but also the toxins that they produce. Also, many of them recognize glycans. Right? So, binding specificity of these glycan binding proteins are important to understand. So, any particular glycan binding protein on a cell, it'd be nice to know what are the structural bits of those glycans that are recognized, and what are the consequences of that binding interaction? Is there any signaling that happens as a result of that interaction? So for instance, a circulating leukocyte interacting with a vascular endothelial cell, we know that there are receptors that mediate those interactions between these cell types. We also know that these binding interactions can lead to cell adhesion. They can also change cellular behaviors in terms of survival, cell division, cellular differentiation, as well as cell migration and other sorts of functions of these cells. This has been very well studied in immunity and in inflammation. To try and capture and understand what the binding specificity of binding proteins, over the last 30 years, glycan microarrays have been developed by many different laboratories and many different well-funded consortiums by NIH and other efforts around the world to build arrays of glycans that can be probed with glycan binding proteins that may have a fluorescent molecule directly attached to them, or can be recognized by secondary reporter proteins with fluorescent molecules. So we can begin to very robustly characterize the binding specificities of glycan binding proteins in a high throughput fashion. And that allows interpreting this data to understand what glycans are recognized, what structural features protein X, glycan binding protein X, may actually recognize in a physiologic context. And it turns out that many of these binding proteins recognize what we call motifs, that is, frequently at the terminal part of the glycan, different structures that can be recognized by glycan binding proteins. Right, so that leads to a generation of a lot of data that's out there now. And this is, I think, really high value data to start to understand something about the biological consequences, to build testable hypotheses, and to also try to interface with other sorts of data, like where are the glycans expressed in different types of tissue? Where are the enzymes that build these glycans expressed? Gene expression data that's out there. Where is the glycan binding protein expressed? And what about the gene that encodes that glycan binding protein? So gene expression, protein expression data. What are the glycoproteins that actually are modified with that glycan that could be recognized by protein expression? So what kind of glycoproteins have that glycan on them? Where is that data? Where would that data be? And where could it be accessed, right? So I've told you already that glycoconjugates regulate essential functions in health and disease. Almost all currently marketed biologic pharmaceuticals are glycoproteins. That the glycan structural features influence glycoconjugate functions. Where is that data? Where is all this data, and how do we harvest it and use it to generate new knowledge? I've also told you that glycans, we represent them with codes of colored shapes, but there are also sort of more harmonized representations that are machine readable, that allows opportunities that you'll hear about in the next couple workshops to begin to do things with that information, right? So there are databases in the world for some of the most important molecules in life, right? So GenBank, RefSeq for nucleic acids, UniProt for protein sequences, lipid maps, and other metabolomic resources for understanding lipid diversity. But of these four major building blocks of life, nucleic acids, proteins, lipids, and glycans, where are the databases for glycans? What about the glycans that define the glycome? Where are those? Well, You're going to hear about what Glycan has to offer over the next couple workshops and appreciate how we organize our data. There are other efforts in the world, GlyConnect, Glycosmos, in Japan and Europe. Together, these efforts, Glygen, Glycosmos, GlyConnect, form the GlySpace Alliance, where we try and make sure that we can all talk to each other and share data as much as we can. And I hope you'll appreciate through these workshops how you can access data and how you can help contribute data to expand. And this is important, and to begin to think about how we can connect Glycan data to other types of data. Right? And so just some of the thoughts that we've had is that, and there are others, but these are sort of some use cases one might think of, that we could link glycan structures and glycan structure expression through gene expression and biosynthesis data, understanding the biosynthetic pathways. Those enzymes are all genes. There's a lot of data out there about gene expression in different tissues and different diseases. And that might be a target for understanding the importance of particular glycan structures and thinking about new targets for therapies. Through glycan binding specificity, I've told you about these array and the array data and understanding what glycan binding proteins exist. I don't think we know what all the glycan binding proteins are, what those binding specificities are, what those known about those proteins in different sorts of disease contexts, and putting together that data to generate new hypotheses. And then finally, I've tried to stress that glycosylation requires a lot of different metabolic input. And so understanding what metabolomic signatures might exist that are associated with particular glycosylation profiles might give us a whole new set of data for understanding how to modulate glycan expression and what it means in a metabolic context. And there are other ways to think about linking glycosylation knowledge to other knowledge. And I hope that some things might come up in these next workshops. And I'm just going to stop there. I just want you to know that in the slide set, which you'll have access to, we'll be posting it, I've also added a bunch of slides that just highlight some of the main points. So you have some quick reference to what I've talked about.